Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, so get this. What if I told you that just a gentle, non-invasive massage on your neck could seriously boost your brain's ability to clear out waste, especially as you get older?
Sounds pretty wild, right? Almost too simple.
Exactly. Today, we're doing a deep dive into this really groundbreaking study just published in Nature. It basically unravels this critical pathway for cerebrospinal fluid drainage, CSF drainage, that we haven't fully appreciated before. And it reveals a, surprisingly simple mechanical way to actually enhance it.
Yeah.
So our mission today is to pull out the most important nuggets from this research for you. We want to show how this, seemingly small discovery could have just massive implications for brain health, for aging, maybe even for tackling neurodegenerative diseases.
It's a really fascinating piece of the puzzle, honestly, and it shifts our whole understanding of brain health in quite a significant way. We often focus on neurons firing complex connections.
Right, the electrical stuff.
Exactly. But the brain's fundamental plumbing system,
Yes.
just as vital. And when we talk about cerebrospinal fluid, CSF, it's not just some passive cushion.
Not just padding.
Not at all. Think of it more like the central nervous system's dedicated cleaning crew. It's constantly circulating, doing its job. Its job is basically picking up and clearing out all sorts of waste. Everything from spent neurotransmitters, regular metabolic junk, to, and this is crucial, those harmful protein clumps like amyloid beta and tau.
Ah, the ones linked to Alzheimer's and stuff.
Precisely. Those infamous culprits. So CSF is essentially your brain's personal, tireless waste disposal system, working 204/7.
And for, decades, figuring out where all that waste actually goes once the CSF picks it up. That's been a bit of a mystery, hasn't it?
That persistent one, yeah. People have been observing CSF drainage since, 1875? But the exact exit routes out of the skull remained kind of hazy. Lots of theories, lots of observations over the years.
And previous research, some really landmark studies using fancy techniques, like PROX1 GFP mice, immunohistochemistry, MRI, they did confirm CSF drains from that fluid space around the brain, the subarachnoid space, to lymph nodes in the head and neck. We knew that much.
We did. But here was the catch. Trying to actually manipulate that drainage, maybe tweak growth factors or even zap certain vessels, it almost always required pretty invasive surgery to get to those deep lymphatics.
Right, you could just, schedule a brain.
Exactly. Not easily, anyway. What's been so frustratingly fascinating is that while we knew CSF ended up in neck lymph nodes, the precise connections, the actual highways it takes, and crucially, how to maybe regulate them without surgery, that was still a major puzzle.
And that puzzle got even more important when we learned CSF clearance slows down as we age.
Yeah, absolutely. That age-related slowdown is a known contributor to impaired brain function, to that protein buildup we mentioned. So finding a non-invasive way to boost this outflow, that's been kind of the holy grail. And that's the core challenge this new research really tackled head-on.
And they tackled it in such a surprising way. This study doesn't just, tweak known pathways. It maps out this specific critical new route for CSF outflow through what they call superficial cervical lymphatics, scLVs. Right there in your neck. So if you imagine your neck has, say, two main lymphatic highways.
Right, interstates, yeah.
You've got the deep ones, the DCLVs, buried deeper, harder to reach. But then you have these more accessible superficial ones, the scLVs.
Yeah.
And this research, it zooms right in on those superficial guys.
Yeah, revealing their surprising importance. And to map these routes precisely, they used these special mice, PROX1 GFP reporter mice. Basically, their lymphatic vessels glow green. It let them literally watch the CSF's journey using fluorescent tracers they put into the brain fluid. And what they found wasn't just one simple tube, but this complex, highly organized network.
Okay.
CSF first enters these initial lymphatics in the meninges, those membranes around the brain, right at the skull base. Then it takes several detours outside the skull before hitting those superficial neck nodes.
So multiple little rivers feeding into these main lymphatic highways. Can you, like, paint a picture of these detours?
That's a good way to put it, multiple entry points. So one route goes through the orbital fissure, that little opening near the eye socket, into what they call periorbital lymphatics.
Okay.
These then connect to scLV1, which drains right to the submandibular lymph node, the SMLN, kind of under your jaw.
Got it. Under the jaw.
Then another big branch starts near the olfactory bulb, brain region for smell.
Right.
From there, the CSF fluid crosses the cribriform plate, think of like a bony sieve in your skull, and joins lymphatics in your nasal lining and sidewall. This stream leads to scLV2, also draining to that same node, the SMLN.
Wait, so fluid from near your smell center goes through a sieve into your nose lymphatics? That's incredibly specific.
It is. And then maybe the most surprising one, they found another branch starting in the lymphatic network of your hard palate, the roof of your mouth.
No way.
Yeah. Connected through little canals, it also drains into scLV2 and then the SMLN. And think about this. The hard palate pathway is really provocative because it's constantly moving, right? When you chew, talk, even baby suckling, that natural motion would constantly be promoting CSF drainage. Just talking or eating might be subtly helping clear your brain.
That is fascinating. So my chewing gum habit might actually be good for something. But seriously, this idea of simple everyday motions being so key makes you wonder what else is going on. And they also noted what doesn't drain CSF, right? Like the parotid node.
Correct. That one handles lymph from facial skin, but not CSF. Important distinction.
Okay, so they mapped these incredibly complex pathways. Then they had to prove CSF was actually flowing through them and how much, right? How did they track that?
Exactly. They moved to direct empirical proof, first in mice, then crucially in monkeys, to check if it applies more broadly. They infused those fluorescent tracers, like TMR dextrin, right into the subarachnoid space of anesthetized mice.
The glowing ones again.
Yep. And then they just watched. They tracked the glow moving through the lymphatics and into the lymph nodes over time.
So what was the big number, the surprising finding from watching that flow?
The big surprise was just how much these superficial pathways handle. Turns out they carry about half of all CSF drainage to the neck.
Half? Really?
Roughly 53% to the superficial nodes, 47% to the deep ones. That's a massive finding. It means these superficial vessels are just as critical as the deep ones we already knew about. Completely changes the picture. And what's more, the system showed this cool adaptability or plasticity. When they deliberately blocked the deep pathways, CSF drainage actually shifted over to the superficial route, kind of picking up the slack.
So the body rerouted it? That's clever.
Very. But the flip side, blocking just the superficial lymphatics significantly cut overall CSF drainage by about 35%. So they're definitely not just a backup plan. They are crucial. Half the team, really.
That's a huge takeaway. And you mentioned monkeys. They checked this in primates, too.
They did. Similar tracer studies in Macaca fascicularis monkeys. And those studies confirmed that these CSF drainage routes via lymphatics are remarkably similar in primates, which strongly suggests relevance to us, to humans.
Okay, that's important. And did they look at the vessels themselves? What are they like physically?
Good question. They found that these scLVs, specifically scLV1 and Linus2, are lined with smooth muscle cells. And these cells form these amazing contractile units called lymphangions.
Lymphangion?
Yeah, think of them like tiny one-way pumps. They actively contract to propel the CSF forward. And these spontaneous contractions were consistent, even when they took the vessels out and studied them ex vivo. So they're definitely not just passive pipes, they're active pumps.
Okay, so active pumps, handling half the load. But here's where it gets really interesting for me. We know CSF outflow slows down with age. About 30% less in older mice. That's a known problem. But why? If these little pumps are still pumping, what's going wrong in the aging brain's plumbing? That's been the big question, right?
That is the million-dollar question the study really sheds light on. And the answer isn't simple, it's multifaceted. Surprisingly, it's not, mainly because the big scLV pumps themselves just, break down or stop contracting spontaneously.
Really? I would have guessed that.
Right, most people would. The lymphangin pumps actually keep functioning relatively normally in the aged mice. The real problems seem to lie further upstream in the collection network and in their molecular signaling.
Upstream.
Meaning, first, there's a reduction in the initial collection points. Aged mice had significantly fewer of those initial lymphatics in places like the nasal mucosa, like 80% fewer. And fewer in the hard palate, too, maybe 10-15% less. Plus, fewer valves in the hard palate lymphatics, those little gates ensuring one-way flow.
So fewer drains for the CSF to even get into the system.
Pretty much. The entry points are reduced. Second, there's this issue with impaired nitric oxide signaling. This is fascinating. Aged mice actually tried to make more of the gene NOS3, which produces the enzyme ENOS needed for nitric oxide.
Okay.
But they ended up with less of the actual ENOS protein. It's like the factory orders more parts, but the final product isn't getting assembled correctly or it's breaking down too fast.
Huh. So the signal is there, but the execution fails.
Exactly. And that leads to dysfunctional nitric oxide signaling. Nitric oxide helps vessels relax and dilate, so if that's impaired, they can't open up as well to let fluid flow efficiently.
Makes sense. And you said a third thing.
Right. Fibrosis. They found increased expression of genes related to scarring and stiffness like postin, COL1A1, in the cells around the scLVs in aged mice. Basically, the surrounding tissue gets stiffer, less pliable.
So it physically squeezes the vessels or makes it harder for them to pump?
That's the idea. It could hinder efficient pumping, so you've got fewer entry points, messed up signaling for relaxation, and stiffer surrounding tissue. It's like the whole system gets gummed up.
Wow. Okay. That paints a picture.
Yeah.
But then the solution they came up with sounds almost sci-fi, but totally practical. This non-invasive mechanical stimulator, a neck cuff thing. Tell me about that. How does it work?
It's really ingenious, right? Building directly on the finding that these crucial vessels are superficial right under the skin, and they have these active pumps. The device applies very controlled, very low magnitude forces. We're talking 0.01 to 0.02 kilograms of force.
Tiny, like a few paper clips worth of pressure.
Exactly. Applied to very specific spots. Around the eye, the side of the nose, and along those scLV pathways. And getting the force right was absolutely critical. They found if they pushed harder, like 0.04 to 0.08 kilogram, it actually constricted the vessels and reduced drainage.
So gentle does it. It's not about squashing them.
Precisely. It's about gently coaxing them, working with their natural pumping action.
And the results when they use this gentle, precise cuff.
Honestly, dramatic. In normal adult mice, this low-magnitude stimulation more than doubled CSF outflow. They measured a 2.3-fold increase in the scLVs themselves and over a 3-fold increase in the downstream lymph node.
Doubled it, just from that gentle stimulation.
Yep. And crucially, it wasn't just pushing fluid around faster. They measured the tracer concentration in the collected CSF, and it was 23% lower. That means that the brain was actually getting rid of more waste, overall increased clearance.
Okay, that's the key. Actual cleaning.
Exactly. And the effect was pretty sustained, too. A single 1-minute session boosted flow for about 5 minutes afterwards, and doing it daily for 4 days kept working just as well.
Wow. And did it connect back to that nitric oxide thing?
It did, interestingly. Blocking nitric oxide synthesis reduced both the natural vessel contractions and how well the mechanical stimulation worked. So NO signaling is definitely part of the mechanism, even with those age-related issues.
That is astounding. Okay, but the biggest question, the ultimate test. Does it work in the aged mice? Can this fancy neck cuff actually reverse that age-related decline in CSF drainage, the very problem we were just talking about?
Yes. And this is the part that's truly a potential game changer. The low-magnitude mechanical stimulation completely reversed the CSF drainage impairment in the aged mice.
Completely.
Pretty much offset the deficit. Tracer accumulation shot up 2.8-fold in the lymph nodes and an incredible 4.7-fold in the scLVs themselves. It effectively canceled out that baseline reduction they see in aging.
That's incredible.
It really is. It means that drop in CSF drainage with age. It's apparently not an irreversible decline. It seems it can be corrected, or at least significantly improved, by simple mechanical stimulation through the skin. It just opens up a whole new way of thinking about maintaining brain health as we get older.
This research feels huge, both technically and conceptually. I mean, technically, you've got this idea of maybe a wearable device for lymphatic stimulation specifically for brain health.
Yeah.
That's a massive leap from invasive stuff.
Absolutely. From benchtop finding to potential therapeutic concept.
And theoretically, it reframes how we think about CSF leaving the brain. It's not just passively oozing out. It's an actively tunable process happening right at the periphery in the neck.
Yeah.
That could really shift the paradigm for brain health, couldn't it?
It really could. This work throws open the doors to potentially new therapeutic avenues for all sorts of conditions linked to bad CSF drainage, age-related cognitive decline, maybe Alzheimer's, TBI, other neurodegenerative disorders where waste buildup is a factor.
Right.
But, of course, it also raises a ton of important questions for the next steps. We still need to nail down the precise mechanisms. How exactly does the mechanical nudge improve things, especially with that wonky nitric oxide signaling in aging?
Mm-hmm.
Can this boost be sustained long term? More than just 4 days, months, years?
Yeah, is it a lasting fix?
And the big one. How do we translate this to people? Human lymphatic anatomy is different, more complex, and we need reliable, non-invasive ways to actually measure CSF drainage in a clinic to see if it's working.
Lots of work still to do, but what a start.
A very promising start indeed.
So this deep dive into this Nature paper really hammers home the importance of these superficial neck lymphatics. It's something we kind of overlooked. And most excitingly, it shows this incredible potential of, well, targeted, gentle mechanical stimulation to maybe reverse age-related brain drainage problems. It's a great reminder that sometimes simple, accessible ideas can have really profound impacts.
And it really makes you wonder, doesn't it? If this gentle, targeted mechanical help works so well, what might that imply about just, everyday movements? Or maybe specific exercises or practices involving the head and neck? Could they be naturally supporting our brain's lymphatic system in ways we don't even realize? What might you, listening now, do differently knowing this connection exists?
Definitely something to think about. Keep diving deep into the science that shapes our world. Until next time.